Hey guys, welcome back to Bioinformatics with R. In this lecture, we're going to be talking about genome synteny and running a script to compare regions of synteny between genomes. So synteny is the idea that organisms, through the process of evolution, conserve things like gene order and these big blocks of genes that they inherited from a common ancestor, right? So you can imagine that if an organism, say a human, has a gene order of a heat shock protein, a hemoglobin protein, and a pigmentation protein in that order, those three, that a chimpanzee or something closely related would likely have the genes in the same order in their genome because there's been no recombination events to break it up and reshuffle. And since these are all physically linked on the same chromosome, that it requires recombination events to shuffle them between closely related organisms. And so this is a way to kind of visualize synteny between organisms. So let's get into it. Okay, so first thing we're going to do is we're going to load the, let's click up here, our script. We're going to load the decipher package and run that. No package called decipher. I've been having this problem a lot, haven't I? BioC, manager, install, some quotations there, decipher. Update all the dependencies. Okay, while that's doing, so what we're going to do here, so in the first step, we load, drag me down there. We load the libraries and the sequence into long seeks. So that's what we're going to call our variable here. And this is I was going to say going to be, but this is a string set object. Okay, so the command for this is we're going to save into our variable that we're calling long seeks read DNA strings set. And for you, again, you're going to use your generic Praxis file destination. If you forgot, it's going to be desktop classroom my files. For me, on my desktop here, I'm going to do file path get wd datasets chapter 3 this was from, and plastid genomes .fa. So we're doing plastid genomes, and this is plastids having much smaller genomes. They're organelles like chloroplast or things like that. Much smaller genomes, much faster computation time compared to doing, a chromosomal or a nuclear genome. So let's read long seek after I run all this. Let's run this. Oh, of course I got to make sure I'm working in the directory that my files are in, so make sure that you're in your my files directory. Now let's try running it. Okay, so you see we have this DNA string object. It's nice and color coded, which is cool, so you can see like cheese are always blue, etc. Alright, so now what we want to do, and you see, let's look at this real quick. So you can see we have a kind of a nested data table here. So now let's build a temporary SQLite database, because that's what we need to use as an input for this package. So we're going to say our seek to DB. So add sequences to a database. So we're going to pull them from that seek, this long seek object that we made. So we're going to say long seek, and then we are going to give it the input type, because this function needs to know what kind of input type. And we're going to say it's an X string set. So this read DNA string set makes these, and this is these DNA string sets. X string set is kind of the parent group. So it's going to ask, so it just knows how to read the data. And then we're going to give it the name of the one we want it to look or to use, which was long seek. And then finally, for the names of the sequences, we're going to tell it to pull the names from long seeks. Long seek. Did I spell this wrong? Seeks to database. Names. There we go. Okay, so we have 5 total sequences in the table, which is great. And so it's this temporary SQL database. So now that we've built the database, we can do the following. And we will find the Synteny blocks. So we'll say our Synteny is our object, is find Synteny from the decipher package in the long database. Oh, hold on here. No such table seeks. What did I do wrong? This is we're loading long seek. It's an X string set. Then we need to say what the name is going to be. It shouldn't be long seek, so we're making long database here. So that's where I messed up. Okay, so change this to long DB instead of long seek. There we go. Okay, so we have this list, this Synteny here, which is great, but you might want to say, all right, well, we can't view it, so we're going to have to view blocks with plotting. So the first thing we're going to do is pairs of Synteny. And we're going to run that. Oh, I spelled this wrong. Synteny. A lot of typos today. All right, now let's run pairs of Synteny. Okay, so you get this comparison of the chromosome of the organism. So these are our 5 organisms, and then how much of it aligns is this kind of direct line here. And so you can see that for the most part we have some pretty close alignments, but for like a comparison, so if you compare, say, this NG chromosome to this HKA chromosome down here, you see that there are conserved regions, but they're kind of all chunked around, right? But then if you were to compare this, the CN, CHLR with the HKA, there's a lot more conservation. So this kind of shows you the how much of the chromosomes are conserved between the different organisms. So we can also do plot Synteny, and you'll get this cool colorful chart here. And so this is treated as our reference genome, and the colors, so you can imagine that this across this entire line here is the base pairs of the plastic genome, right? And so the conserved chunks here, these blocks, are conserved regions. So as you can see, between the SJ chloroplast and the NG chloroplast, we have a giant region that shows high conservation. And then between the SJ and the CN chloroplast, we have a conserved region, a pretty good sized one, but the difference between SJ chloroplast and the AN chloroplast, there are some conserved regions, but it's much more broken up. So if we were to go back to our previous graph, so between SJ and what was the other one, NG, you see that there's some big chunks here that are missing, and so this was the least conserved among the groups, right? Let me double check. It was SJ and AC. Is that what I said? Between SJ and AC, yeah. So that kind of just gives us an idea. You know, I'm not going to get terribly into the weeds. Like I said, this should be a entire lecture series. But the other thing too is for that heat map. So here we have pairwise comparisons between everybody, right? So we see the comparison between this SJ chromosome and the ANCL, or the AN chloroplast, and the NG chloroplast, and the CN chloroplast, and this HKA. But when we do we also have comparisons between the AN and the NG, and the NG and the CN, etc. So we have all possible pairs compared to each other. When we do the actual this heat map, this only compares to one reference, right? So this is only comparing AN, NG, CN, and HKA back to the SJ. So it all depends on these the order that you put them in when you create this database. So if you wanted to compare CN to NG as the reference, then you'd have to reorder them. Okay, so now let's make an actual alignment file. So we're going to call this alignment. Oops. And we'll say align Synteny for alignment for 1. So what kind of stinks about this is oh no, it's for Synteny. Oh no. Oh, I'm mixing up my commands here. All right, for our alignment, it's going to be align Synteny, which is a pairwise alignment of all blocks of Synteny between sets of sequences. And so we have to give it Synteny is what we're going to use. Synteny, and from our long database. Okay, and now let's create a structure holding all aligned syntenic blocks for a pair of sequences. So with this package, besides that table, we have to export each comparison by itself. You could write a function to go through and do them all, I suppose. But we will just do the first one. Did I not run this? Oh, there we go. Takes minutes, a lot of comparisons. Okay, so now we're gonna run this block. So we got former class. This is a DNA string set that we had worked with in a couple of the other videos. So we can write to file one alignment at a time. So we can do write x string set. So this uses is able to read the string set data structure and write out to a we can write out to a FASTA file. So we can write this. And so in your working directory, you should have, what is this one called? Synteny. This FA file, you should be able to open it with Notepad, that has the alignment. And so the dashes means that there are no alignment at that position. And that's like gaps. So these are the actual places that do align to our reference where there are letters, and then the gaps, there's no alignment. So let's get back out of this. So that was just a brief taste, little teaser of doing some Synteny stuff. You could write your whole PhD thesis on studying synteny between organisms. But I just want to expose you guys to the things that you can do with R. You're not going to, at the end of this course, you're not going to be an expert in every single thing of these, because, you know, parameters can change depending on the analyses you run. But I want to give you guys some examples of what you can do outside of RNA-seq. So that was a taste of Synteny, but you can get much more complex and look at, you know, there's things called CNSs, which are conserved non-coding sequences, which are really interesting because you would think that genes are conserved because there's evolutionary pressure on these genes to, if they break, that organism would die. But if there's a sequence that is non-coding, it's not making a protein, and it is conserved throughout evolution, why is that conserved? So there's got to be some sort of importance to it, right? Because if there's no evolutionary pressure, you should get mutations there and it not be a big deal, but they are conserved regardless, and so CNSs are an important aspect. They're something that you could study with the Synteny to figure out why these non-coding sequences are there. But that's neither here nor there. This one's been a little bit of a mess, hasn't it? Isaac's shaking his head. So that's it for this lecture. I want to keep it somewhat short. This is already longer than I thought. I hope you enjoyed, and I'll catch you guys in the next one.